So now we're going to look at the physiology of the hippocampus and see if we can see evidence of encoding of time. So if you remember, we've categorized time in two different ways. One is according to the type of time that we're interested in. Is it exactly how long ago something happened, or is it the order in which events happened, or is it the time of day at which something happened? And we've also looked at the different time scales. So there are millisecond time scales, seconds, minutes, days, and so on and so on. And so when we look in the hippocampus, we see phenomena at each of these different scales. So, for example, we've met millisecond scale timing in two scenarios in the hippocampus. One is spike timing dependent plasticity. So if you think back to the lectures on LTP, we saw that the exact time of arrival of the depolarization of the presynaptic versus the postsynaptic neuron would determine whether that synapse would get stronger. And so that's a way of building connections that are in the direction that information is trying to flow, as it were. So that's one type of millisecond scale timing. The other is in theta, and although theta itself is a phenomenon that unfolds over fractions of a second, so tens or hundreds of milliseconds, there are actually phenomena within a theta cycle that operate on the millisecond time scale. And so we've met phase precession, and one of the reasons that phase precession is interesting is because the very precise timing that it allows might be a way of allowing the hippocampus to learn sequences. So we've just seen that the hippocampus seems to be important for learning sequences of events and sequences of places and all sorts of other things. So how might this work? So the basic idea is this. So imagine that you've got a rat walking along a linear track and it's going through a succession of place fields, one after the other. So you can see those as the different coloured tuning curves, if you like, of firing rates of each place cell, sort of red and orange and yellow and so on. And then the trace underneath is showing you the a sort of stylized theta wave. And we can imagine that because of theta phase precession, you get a particular pattern of organization of the spikes of those different place fields that reflects the spatial organization of those same fields. So, for example, if you look at the red cell and you look where it's firing on the theta rhythm, you see that the very first spike, when the rat first entered the place field, coming in from the left, as it were, is just before the peak of the theta wave. The second one is further before the peak. The third one, and I've shown it slightly broader to reflect the increased intensity of firing, is near the trough, the preceding trough, and the fourth one is on the sort of descending phase of the theta wave. So at each successive cycle of theta, the firing of that cell is earlier and earlier in the cycle. Now, by the time the rat has got to the second theta cycle, it started to enter the beginning of the orange place field. And so the orange place cell will start to fire. And again, it will start to fire just before the peak. Meanwhile, the red cell is on its second burst of firing, and it's firing slightly earlier. So if you look at that theta wave, the red cell is firing first, and then the orange cell is firing. And then the rat carries on a bit more. Now it reaches the beginning of the yellow place field. So the yellow cell starts to fire just before the peak. Meanwhile, the orange one has precessed slightly earlier and fires just before the yellow one. And the red cell has precessed even earlier and is firing before the orange one. And so on and so on. So by the time the rat has got to the middle, and you can see the little cutout there, if you look within that theta cycle, you can see that the red cell fires first, then the orange one, then the yellow one, then the green one, then the blue one. It hasn't reached the purple one yet. So within that one theta cycle, you have a little recapitulation of the spatial sequence of that entire set of place fields, even though that entire set of place fields is quite sort of distributed. So what that's done is that's temporarily compressed the spatial organization of those place fields. And it's meant that those cells are firing within a few sort of tens of milliseconds of each other, or even less, a few milliseconds. And that starts to meet the conditions under which plasticity can occur. So that has brought the firing of those cells to within the temporal range that will allow them to build connections, and then subsequently those cells will be able to fire in a sequence, and we'll get things like replay of those sequences and so on. So there's reason to think, and this is still hypothetical and we still don't know for sure, but there's reason to think that the phase precession might be a timing mechanism for organising spatial sequences or spatiotemporal sequences. So it's also the case that the hippocampus is showing phenomena that are operating on the scale, on the mesoscale, the scale of seconds rather than milliseconds. And this is a set of experiments that have revealed the existence of what we have come to call time cells, although it's a little controversial still exactly what they're encoding. So one of the first illustrations of this was this study by Pastelkova and colleagues. And so what she was doing is looking at what happens when a rat is paused in a dynamic state in a task that it's doing. What's happening to its place cells while it's not moving through space, but it's essentially moving through time. So what happened was that the rat had to do this sort of figure of 8 task where it was running around a track and place cells were being recorded, and you can see the cells color-coded in the usual way. And on each lap of the track, when the rat got back to the start, it was confined in a running wheel, and it had to run on the spot for a certain amount of time, and then it was released, and then it would go and do another lap of the track. And the interesting observation was made during the time when the rat was in the running wheel. So it's not moving through space. But what she found was that place cells nevertheless would become active, and they would become active in sequences that reflect the sort of sequences that they appear in the track, and at specific times during the time interval. So you can see some examples on the bottom left there of, so each row in that block is showing the period of time and the firing rate. So it's a heat plot of firing rate, and you can see for a given cell, which is one of those panels, the firing rate increases at a specific time within that time interval, and it's a different time for each cell, and it's very kind of specific. And on the right you can see all of the cells that are arranged in order, progressive order of the time at which they became active, and you can see that the time for which they are active is quite well circumscribed and pretty reproducible. So the consequence is that these cells are not just firing at specific times, but also in specific sequences. So a similar phenomenon was reported not long after for another task in which a rat had to wait for a period of time, but this time it's not running. So the only thing that's happening during that interval is that time is passing. So it's another one of these linear track tasks. In this case, the rat had a memory task to perform. So what happened is it would be presented with an object in front of a barrier, and it had to learn the association of that object with an odour. So the rat had learned a set of object-odour pairs. Then the barrier was removed and the rat ran into an enclosed space. The barrier was then restored behind the rat. So now it's confined in a space on the track for some period of time, so typically 10 seconds. And then at the end of that time, the barrier in front of it was removed, and it would be confronted with a pot of sand that was scented with an odour. And if the odour was the one that was paired with the object, then the rat would dig and get a reward. And if it wasn't paired with the object, then there would be no reward. So the rat is having to wait for this 10-second period of time, holding on to something in its memory, and then it runs around the track and the sequence repeats. And what they found is that, again, during that delay period when the rat is confined, not moving on the track, there were nevertheless cells, which in all other respects look like place cells, and they would become active at specific times in that time interval. And you can see the raster plots, the middle panels are the recordings that were made during that delay period, and you can see that for these 7 cells, each cell has a specific period within that 10 seconds that it becomes active, and as with the previous experiment, that period is the same on each trial and individual to a cell. So we've taken to calling these periods of time time fields. It's very analogous to place fields, except occurring in time. And they have certain properties that are similar to place fields. So, for example, if you change the context, then the time cells remap. If you expand or compress the time, the waiting time, then the fields expand or compress as well. So it's been suggested that these hippocampal cells are not just place cells, they're really place time cells. They are encoding, if you like, 4 dimensions of space-time. And that is potentially a mechanism for the encoding of sequences that we've seen that the hippocampus is necessary for. So those are sort of micro and meso scales. Is the hippocampus interested in macro scales? So there's reason a priori to think that it would be because of its involvement in episodic memory. But it's been somewhat difficult to show. But there have been some experiments that suggest that it might be encoding long scale time, but in a different way. So this is an experiment that was done shortly after development of a new recording method, which is microendoscopy. And with microendoscopy, you attach onto the rat a tiny microscope that can image the calcium activity of genetically encoded calcium indicators. So these are transgenic mice that are expressing a protein that fluoresces when calcium flows into the cell. So they're activity markers. And you can see this fluorescence if you look under a fluorescent microscope. So this imaging method allows the simultaneous imaging of lots and lots of cells at once, and it can be done for long periods of time, like days or weeks. So in this experiment, what they did was implant one of these microendoscopes. You can see a diagram of it on the left, and in the middle you can see the image field. So each of those orange dots is an active cell, and then on the right are the signals, the calcium signals. So these are not action potentials. They are slower than action potentials. These are calcium flowing into the cells. But they somewhat conform to place fields. So when a place cell is very, very active and firing lots of action potentials, there'll be a big spike in the calcium in that cell. So these calcium indicators can tell you where the place fields are. And what was found is that if you record from these cells day after day after day, you find that there's a subpopulation that maintain the same place fields and another population that slowly turn over. So if you look at the first panel on the bottom there, that's showing day 5. So each of those 178 cells is arranged in the order of the location on the linear track where the cell is active. So you can see that the heat plot shows this sort of rightwards progression because it's been aligned that way by hand. But then if you look at the subsequent days 10, 15, 20 and so on, you can see a ghost of that same diagonal stripe. So that's from cells that have maintained the same place field even after all of those days. But the stripe is not a perfect reproduction of the stripe on day 5 because some of the cells have remapped. So there's been a turnover. Some of the cells are stable and some of the cells have changed their firing. And the more time goes by, the fewer cells have maintained their firing and the more cells are firing differently. Now, it's not that the recordings are unstable because the stable cells tell us that the microscope is still seeing the same set of cells. They're still appearing in the window in the same way. What it seems to suggest is that the activity of the place cells is slowly kind of percolating, if you like. And it's been suggested that this might be a mechanism for comparing the temporal proximity of memories. So if something happens and then another thing happens, if those two events are close in time, then the associated place cell population will be very similar. But if they are in the same place but separated by a period of time, like a week later or 2 weeks later or a month later, then the population of place cells that are simultaneously active at the time of that event will be different. And the more different they are, the more time must have elapsed. So this could be a way of allowing the brain to compare the age of memories. So we don't know if this is true. It's still speculative at this stage, but it certainly shows that there are processes in the hippocampus that are operating on this sort of macro time scale. Now, I've mentioned that circadian timing is not the function of the hippocampus, it's the function of the suprachiasmatic nucleus. But there have been some suggestions that place cells do show a circadian cycling, if you like, that's similar to this long-scale cycling that I've just told you about, except it's on a 24-hour cycle. So this is an experiment where they recorded place cells in two types of apparatus, a circular apparatus or a square apparatus, and they recorded either in the morning or the afternoon, and they recorded in the CA3 region of the hippocampus or the CA1 region of the hippocampus. And what they found was that when they compared the patterns in the morning and the afternoon, the patterns were different in CA1 but not in CA3. And then the patterns of the successive mornings would be more similar to each other again. So there seemed to be a sort of a turnover in the CA1 but not in the CA3 population. And this is potentially a mechanism for allowing the brain to learn the time of day that certain events occur. Now, in rats, it's been quite difficult to show that rats actually can link events to the time of day. But it seems likely that they probably can. We haven't just found the right way of the right behavioural task to elicit that yet. And it may well be that this hippocampal circadian cycling of place field location or place field similarity is a mechanism for that. So we now have in the hippocampus a mechanism for tracking time of different scales, and the question now is, how does that pertain to episodic memory? And we're going to look at that next.